I would love to introduce our next speaker. His name is Dr. Mohammed, and we are going to be talking today about the application of functional lifestyle genomics to autistic spectrum disorder. And, I know that's a lot to take in just that sentence alone, but I promise you that before this hour is up, you're going to have a whole new idea about the genetics and how that relates to autism. So thank you for being here, Dr. Mohammed.
It's a pleasure and honor, Kristen.
And so before we get started on the subject that I know so many families are listening and I'm so excited for them to be able to hear you speak, but I know that they're going to be excited to learn. Can you give them a little bit of background about why you're so passionate about what you're doing?
Sure, absolutely. I'm a clinical genomicist. I did my first couple of postdocs at UCLA and then my final postdoc at Baylor College of Medicine. And those were sort of the heady days of the genome, the Human Genome Project. It was sort of when we were finally getting a scope of just how important genetics was. It was coming to life. The fruition of genetic testing was really coming through. And I was just simply lucky and blessed with being at the right place at the right time with mentors both at UCLA and Baylor College of Medicine. But one of the things that struck me was when it comes to DNA testing, I think most people, we understand DNA and we tend to understand genetics in one of two ways. We tend to understand, the average person will understand the genetics relates to their outward appearances. Why is my nose more like mom and my chin more like dad? We understand that and we have an intuitive sense of that. And then on the other side of the spectrum, we tend to think of genetics when it comes to disease, bona fide mutations and cancers and diseases. But what we forget is the genetics that falls in everything in between, i.e. beyond the superficial external appearances and well before it's an actual mutation that causes a disease. In other words, our genetics makes up and determines to a large degree how our body functions. And there are many variations to that genetic makeup that then alter the way our bodies function. And so that's where my passion lies, in understanding this thing, this operating manual of the human being, understanding that we have access to this operating manual, and that we can read this operating manual, at least, there are those who can intelligently, and that we can understand the individuality, the uniqueness of the person through their operating manual, and then from that, how are they interacting with their environment, their nutrition, their lifestyle, and how does it all come together to impact on that person. So that field of functional genomics, lifestyle genomics, is where I've now put my emphasis. I spent many years in the cancer diagnostics world and then in the childhood developmental world, both prenatal and postnatal. But I've really put my passion now to understanding, just truly stepping back and saying, how can we understand the human operating manual, our genetic makeup? Does that seem to make sense?
Absolutely. I think that so many people want to understand it more, and today there's different kits that are out there, and they give you little bits and pieces of who you are, and I think we don't really understand it still. But I love the fact that the science is here, and there's doctors such as yourself that are really just bringing light to this and really explaining it in terms that families like the ones listening and myself can understand. So let's jump in and, what is functional and lifestyle genomics?
It is, as noted, it is the study of this operating manual. It's the appreciation, Kristen, that if here we are, two human beings, if we went out and had a lunch together now, and we ordered the identical meal, literally the identical meal, and we completed that meal in its entirety, each of us individually, and we, after that meal, had a cup of coffee, which we drank to entirety, having had the same cup, sweetened the same and everything else. And while we were having that cup of coffee, we were in a patio being exposed to someone who was smoking next to us. So just the general, pollution or whatever else might be in the environment. As two human beings, despite that previous 45 minutes that we were together, despite our bodies experiencing the same vis-a-vis the nutrition we entered into our bodies, vis-a-vis the environment we were sitting in, An hour later, as you go back to your office, I go back to mine, what is happening in your body, how your body is going through the steps of digesting, absorbing, metabolizing the nutrients in the food that you ate, detoxifying your body in the pesticides that we, neither of us, were aware of, that were residual on the vegetables that were in the food that we ate, how your body detoxified itself from the smoke that both of us were exposed to. This is my body. In other words, here we are, two human beings, but at the minutiae, at the cellular level, how our body operates can be radically different. And this difference and the understanding of this difference through the lenses of genetics is what functional and lifestyle genetics is about.
Now, how is functional and lifestyle genomics different from current approaches, to genetic testing? Because we all have heard of, like 23andMe and things of that nature.
One of the ways I like to put this is, I tell the story of the fallacy of clinical trials. And it's a well-documented concept. And it's important that our families, and this is what makes me so passionate about this, it's important we understand this. And it goes like this. So a freshly graduated mechanic, young man or woman who's just finished mechanics school, he's gotten his first job, and he goes to the shop, and waiting for him at the shop are 100 cars that cannot start. In other words, 100 cars waiting in the parking lot that could not start on his first day, and he or she is told, well, fix the cars. And so he remembers in his course, well, the starting box, and I'm dating myself now because I guess maybe new cars no longer have starting boxes, but you'll understand what I mean. He remembers from the course that he took that the starting box is necessary for cars to start. So what does he do? He gets 100 new starting boxes, goes out to the 100 cars, and he replaces the starting boxes. Okay, so he's done that, and then he tries to start the cars. And once he does that, he finds of those 100 cars, only 5 of them have been fixed. Only 5 of them are now working. So what does he do? He comes to the conclusion that because 5 out of 100 is not statistically significant in his way of doing math, he says starting boxes are not relevant or not important to starting cars. So you see what he's done there is he has taken something that most certainly is necessary for starting a car. He replaced all 100, finds that only 5 of the cars were solved in terms of fixing the problem, and he draws the erroneous conclusion that starting boxes are not necessary for cars to start. This is the fallacy of clinical trials, which is not to say that clinical trials don't work, but it is to say that when we approach the human body as single gene events, without taking into context the bigger picture, without taking into context how genes interact and work together to bring about the whole, when we neglect that, when we do not look at the bigger picture, we often lose so much. So in this case, this young man or woman walks away thinking, erroneously, obviously, that starting boxes are no longer important for cars. It just happened that the other 95 cars that didn't get fixed, the starting box was not the problem at hand. It doesn't mean that the starting box wasn't important. And so functional genomics looks at genomics in a slightly different way. As functional genomicists, we're always watching out for the pathways. We're always watching out for the interconnectivities and the systems in the body, making sure that we're not taking this singular gene event. And I'll close this answer by saying, I like to make the analogy that we're not, if you've got an orchestra, a well-played orchestra isn't different musicians with different musical instruments playing to their own rhythm. A well-played orchestra is where it all comes together, playing together in a simultaneous, in a well-rehearsed manner. And that's the nature of genetics in the human being. It's very hard to draw conclusions based on single genes. Not saying that single genes aren't important, but we've always got to keep in mind the bigger picture. That's the difference between a good functional genomic evaluation and a sort of singleton gene approach. The latter, a lot can be lost and a lot can be misinterpreted taking the single gene approach. Again, does that make sense?
Oh, yeah, absolutely, because I feel like, and this is basically, I guess, probably a really basic question that people like you know, but I guarantee you many of our listeners don't know, and I know I don't know. How many genes do we have in our body? I know there's just so many, but how many are there?
So in terms of actual genes, meaning discrete instructions, we now believe it's somewhere between 20 to 22,000 genes. And notice I'm still saying somewhere between, meaning even, you'll hear folks saying, well, we've sequenced the human genome. We have sequenced the human genome by and large, but we're still trying to determine the exact number of genes. When one gets to understand the complexity of these genes, you see, if your genetic makeup was a book, an instructional manual, as I keep using the analogy, the way we understand genetic manuals or books is that a book is divided into chapters, and good enough, we can use the analogy that our instructional manual is divided into 23 chapters, corresponding to the 23 chromosomes, of which, by the way, we have 2 copies of. So one of the unique and miraculous and beautiful things of the human genetic makeup is we have 2 copies of our instructional manual. One we receive from mom, one we receive from dad. And both of those copies are by and large identical. By and large, obviously they're not completely identical. Each has 23 chapters. Now within those chapters, as chapters are wont, you're going to have opening paragraphs, sentences, and words. In the simple way of understanding a book, we tend to think of, well, we start reading, and we have chapter 1, paragraph 2, paragraph 3, and so forth. When we apply that analogy, it's a very good analogy to the human genome. However, the human genome does not work in the way of just purely discrete paragraphs, in which case each paragraph can be considered to be a gene, in which case we have between 20 to 22,000 paragraphs or genes. The reason for that is the way the human genetic structure is, the architecture behind our DNA, our genetic makeup, is that sometimes one paragraph is a particular gene, a particular instruction, but then that same paragraph can make a part of another gene. So in other words, a single paragraph may not just be one gene. So that probably is a little too much detail. Suffice it to say, we believe we have somewhere between 20 to 22,000 genes, individual, discrete instructions that collectively are what are needed to give rise to what we see as the beautiful human form and function.
Well, I feel like that is a great explanation to so many families listening. And, when you're talking, what is the difference when you're thinking about maybe between diagnostic and predisposition associated with genetic testing?
An excellent question. Again, this is a little bit of that historical note that I mentioned earlier. When we think of genetic testing, because for good reason, a lot of genetic testing came to light and has been used and continue to be used in the realm of cancer diagnostics, we tend to think of genetic testing as, if I have such and such DNA sequence change, then I have such and such outcome. So a good example of this is, if a person has not 2, but 3 copies of the entire chromosome 21, that person will have Down syndrome. In other words, trisomy chromosome 21 is diagnostic of Down syndrome. So this is a diagnostic. If you have the change, you will have the outcome. Okay? And we've gotten accustomed to that terminology. Okay? However, human function, form, function, and the things that can go awry, the things that can go wrong, are well beyond a diagnostic change. In other words, there can be changes to our genetic makeup. Each individual genetic change does not necessarily spell out an actual outcome, an actual disease state, but it increases the disposition toward that disease state. And then other factors such as lifestyle, environment, nutrition will be the things that push us over the limit and causes that disease state or that disorder or that malfunction in the body to transpire. And there are many excellent examples of this, which I hope we'll cover some of them as it relates to autism a little later in our discussion.
Now, what is the difference between a mutation and, a polymorphism?
Mm-hmm. Another brilliant point, and it's one of my pet peeves, and obviously being a genomicist, we tend to split hairs over this, but I think it is important. It's important, certainly in the medical community, that we get our terminologies correct, so that even in the overall population, we're not giving mixed signals. So how many times do I hear a person or a family says, yes, my child carries the MTHFR mutation? The famous and well-documented MTHFR gene, the methylene tetrahydrofolate reductase gene, the variation within that gene is not what we call a mutation. Correctly speaking, a mutation will be a genetic change. It's the same concept, it's the same phenomena, but we will only correctly use the term mutation when that change to the genetic sequence is so rare that it is, quote-unquote, not found in the average population. If that change, that genetic change, is rare enough that it is not, again, quote-unquote, found in the average population, then we will call it a mutation. If, on the other hand, that genetic change, that variation, is found in, even if it's to a low degree, but is found in the average population, we will call that a polymorphism, meaning that it is an accepted variation, a variation that does not necessarily bring with it a disease outcome. When we use the term mutation, we tend to, in the proper context, mean that that gene variation necessarily brings with it a disease outcome. So let's take very quickly the MTHFR gene. The famous C677T variation means that at the 677th position within the MTHFR gene, we have found that the sequence, the hundreds and thousands of DNA bits or nucleotides, letters, the A's, C's, T's, and G's that make up DNA, we have found that at the 677th position, we've documented that it can either be a C or a T. Now, we've got two copies of that gene, one from mom, one from dad. And what we found is the more common occurrence is that an individual will have two of the C versions of that gene, one C version from mom, one C version from dad. Now, when you have the two C versions at the 677th position within this gene, that is considered the optimal or most functional version of that gene. For every T, if one of those C's, instead of being the C version, you have the T version, then the ultimate outcome, what is the outcome of this gene? A particular enzyme, the MTHFR gene, encodes the MTHFR enzyme, which has a very important function within our cells. And if one of your two versions is a T, then the enzyme that is produced has a lower efficiency. It's not doing the job that it's meant to do as well. And of course, if you have two T's instead of two C's, then the enzyme is working even less optimally. Now, the fact of the matter, though, is there are many people that have the two T's and they're perfectly fine. So this is not a mutation, it's rather a polymorphism. It's a variation that occurs in the human being, in the human public, in human beings. But by having those T's, either one or certainly two, and with the reduced function of the MTHFR enzyme that the gene encodes, now we are increasing the risk for certain things to go awry, for certain things to misbehave within our cells. It does not diagnose the disease, but it can predict, it can predispose to certain things going off track. This is the difference between mutation and polymorphism.
Now, clarifying the term SNP?
So an SNV. So again, another pet peeve of mine, many people, when we talk about genetic variations, we correctly, the first things that come to mind are what we call SNPs, which is, in the case of the MTHFR gene and that famous C677T variation, this is a SNP. A SNP stands for a single nucleotide polymorphism. Every one of those little letters, every one of those units in DNA is known as a nucleotide. That's the letter of the DNA, the alphabet of the DNA. And the alphabet of the DNA is made up of C's, T's, A's, and G's. Those letters obviously simply represent the letter that designates the fuller name of each of those nucleotides. So we'll stick with C, T's, A's, and G's. When one of those letters are changed, such as from a C to a T, in the example of the MTHFR variant, the 677 variant, that's a single nucleotide polymorphism, or SNP. And definitely SNPs have been well documented to contribute. We each have thousands upon thousands of SNPs that make us and that contribute to what makes us unique. So SNPs collectively are a huge component of what makes an individual unique. However, SNPs are by no means the only variation within your genetic makeup that makes you unique. You see, we have noted that a person should have 2 copies of every gene, one from mom, one from dad. Everyone has 2 copies of the MTHFR gene. And then the only question would be, of the 2 copies you have, do you have the C versions, the T versions, or one C and one T? Whereas, what we've discovered, and that's work that I did with my colleagues, Dr. Steve Scherer at Sick Kids Hospital, University of Toronto, and Dr. Charles Lee, previously at Harvard, and he now has moved on. I think he's the director of the Jackson Institute in the U.S. now. It's been some time since we've touched bases. The work that we originally did, Kristen, was we actually found that every average individual, every human being, there are places in our genetic manual where we don't have 2 copies of the gene in question. You see, we were supposed to have 2 copies, but believe it or not, Kristen, every single one of us is walking around with examples in our dual operating manual where there are paragraphs missing. We may only have 1 copy of the gene in question, or believe it or not, Kristen, we may not even have the gene in question at all.
Wow.
Or versus that, we may have more than 2 copies of the gene. This phenomena where an entire gene, not a letter within the gene, but the entire gene is deleted, or we have more copies than 2, is known as copy number variation, or CNVs. And CNVs, as you might now imagine, given that we all have them, and given that CNVs can literally mean that we don't have a gene, or that we have more than 2 copies of that gene, CNVs have now been documented through hundreds of publications, to be an even greater contributor to the genetic variability of one person to the next. And so when I hear people only talking about SNPs as the contributing factor in genetic variability, or the contributing factor to what might lead to syndromes, including the autistic spectrum, it bothers me, because CNVs, copy number variations, are as much, if not more, a contributor, and no one seems to be talking about it.
Well, now what is the role of methylation then, when you're thinking of all the SNPs and the CNVs and,
Indeed.
Yes.
So what we do is, first and foremost, our entire manual from start to finish, these 23 chapters, of which we have two of the 23 chapters, from the very first to the very last of the chapters, inundated throughout it are SNPs. Inundated throughout it are these single letter changes or single nucleotide polymorphisms. The CNVs, what we have found, because they're so potent, they can literally determine whether you have a gene or not, or you have more than one copy of a gene. We tend to find that these CNVs are isolated to parts of the genome, that parts of your genetic makeup that seem to be the instructions that are adaptable. And by that I mean CNVs tend to, this is a broad statement, tend to be in parts of our genome where the instructions were things that, our body adapted to through the years. So very quickly, very, very quickly, we have the gene that encodes salivary amylase. Salivary amylase is the enzyme in our saliva that breaks down starchy foods, right? So as we begin to chew that slice of bread, the pasta, the rice that we're eating, in our saliva is an enzyme, amylase, that is breaking down said carbohydrates and starches. Well, we would think that we should have two copies of that gene, the gene that makes the enzyme. We should have one copy from mom, one copy from dad. It turns out that the average individual has not two copies of that gene, but 6 or so copies of that gene. That's the average individual. In other words, over a millennia, as we adapted to eating more starchy diets, people, as human beings, we ended up with more copies of that gene in question. And actually, the Yoruba Africans, a wonderful, noble tribe in Africa, whose diet is almost entirely starchy foods, they don't have 6 copies of the gene. They've got 18 copies of the gene and even more. So what I'm saying here is that CNVs tend to cluster in regions where we can sort of see these strong adaptation qualities in the body, including things like how the body detoxifies itself. So within the methylation cycle, the wonderful genes, not one gene, not just MTHFR, but multiple genes that work in this orchestral masterpiece to bring about this thing called methylation, we find that these genes are not affected by CNVs, but they are affected by SNPs. Now, other important pathways in the body, particularly one of the ones that I'm very interested in, and that is how the body detoxifies itself. Those genes involved in detoxification are susceptible to the phenomena of CNVs. And so to answer your question, in short, in the methylation genes, we don't find CNVs, we find SNPs. But in other important systems and pathways of the body, we find radically informative CNVs that can hugely make divergent one person's detox capacity versus another.
That is so fascinating. And, you brought up, the MTHFR, and I know many families that have been on this journey for a bit, have heard MTHFR. So you spoke a little bit about it, but how do we really think beyond that? Indeed.
So MTHFR is certainly one of the cornerstone genes in a process in our cells. All of our cells practically have to be able to do this process, and it is the process of methylation. So methylation is a complex biochemical pathway, meaning several different steps, and methylation has to happen efficiently in our cells. Methylation is important in several things in the body. Methylation, this collective biochemical pathway, is important in, for example, how our neurons function, how neurochemicals in the brain are metabolized, how reproductive hormones, steroidal hormones are metabolized, how it's also important in detoxifying the cells. It is important in how DNA itself is kept vis-a-vis methylating DNA or not methylating DNA. So methylation is what I call one of these master control biochemical reactions in the body as a whole. Now, once we understand the importance of the biochemical reaction, the multifaceted, multi-stepped methylation reaction, it's then important for us to understand that MTHFR and the gene MTHFR is only responsible for one of the steps within this beautiful cycle, this beautiful biochemical cascade that is known as methylation. And so yes, the MTHFR gene is important, and yes, the variation within the MTHFR gene, or variations, of which the 677 variation is one of the most important, yes, when you carry the T allele at that 677 position instead of the C allele, your enzyme, your MTHFR enzyme, is working less efficiently to accomplish the step in the otherwise cascade, and so it will be important. However, there are other genes in that cascade. There are genes before the MTHFR gene, such as the SHMT1 gene, and there are genes after the MTHFR gene. So to simplify methylation down to one gene, MTHFR, is a gross oversimplification, and to only look at that gene is to miss so many other important aspects, because ultimately it's not the gene that is important here. It is the ultimate outcome of methylation as a whole. And whatever affects that methylation, whatever diminishes the efficacy of that methylation is what matters, whether it's MTHFR, whether it's SHMT1, whether it's MTR, whether it's MTRR, and I can go on listing all of the genes involved in that pathway. So again, in conclusion, as I started, in functional genomics, we are not losing sight of the big picture for a single pixel. We are looking at the pixels, yes, we're looking at the genes, yes, but we're always stepping back and understanding how that gene works in the big picture so that we're not hung up on just one gene at the expense of other genes. Does that make sense?
Yes, absolutely. So when, it's interesting because it feels like that's all we really hear about in autism is the MTHFR. I mean, I'm sure they must be talking about other stuff, but I feel like that's the one we always remember and think about,
It really, that's I think, Kristen, when we first met, and hence this talk, it's one of, genomicists whose life's work is on this. It's one of our pet peeves that when we hear these things, and that is not to say MTHFR isn't important, okay? I mean, it's obvious to all of our wonderful families that children affected within the autism spectrum disorder, there are clearly variations, there are clearly degrees of severity of the presentation. And clearly, looking at a single gene, MTHFR, and I noted to you, there are many, many people that are absolutely what would appear to be functionally normal, functionally, no quote-unquote obvious signs of dysfunction that carry the T allele of the MTHFR 677 variation. So what we have to understand is there are other significant variations, and that what we see in our beautiful autistic children as the outcome, that physiologic outcome, behavioral, physiologic outcome, is the sum total of things that transpired both at the genetic level in many cases, as well as then that genetic level complicated or the threshold having been crossed because of environmental, lifestyle, nutritional shifts that happened at radically important times of brain development. So we have to look at the whole, and we can't get hung up on single genes.
And, for the families listening, especially the ones that are, doing the protocols for the MTHFR, so if they are positive, a good portion of the kids, it seems that they have this MTHFR gene that turns out positive, right? Mm-hmm. So for those that are doing the protocols, are there other steps that a doctor like yourself could say, okay, well, now we know that's positive, but did we look at this or did we look at that? Because this may also be a contributing factor that potentially could to help, that child even develop even better or progress more.
Absolutely, Kristen. So, again, we always must step back and we must always ask the question, what has transpired? What maybe was not fully functional, or the unfortunate converging of circumstances where we had a genetic predisposition together with a particular environmental exposure. And oftentimes you see this is where we are missing observations. This is that fallacy of clinical trials example that I gave earlier. Because, there are many naysayers out there that will talk about, well, such and such environmental exposure is not associated with autism. Just look at the statistics. It's not. I'm not saying that there aren't things that we may have overemphasized as causative factors in autism. There may be. But there are certainly things that have been minimized by certain aspects of the scientific community, because they use statistics to say, if such and such causes autism, then why don't all these other children have it? And the simple answer to that is in that example I gave of the fallacy of clinical trials, which is, for the child in question, if we had an environmental insult, for example, and whether that is an environmental insult that affected the child in utero through a microbial infection, through an environmental exposure, through something else, and then the toxin in question, when that child is faced with that toxin, again, whether it's prenatal and or postnatal exposure, then if that toxin is a toxin that can affect neurologic development or any number of other systems in the body, and if that toxin, or I should say, and if that child has exceedingly poor, documentedly, genetically, exceedingly poor, innate genetic capacity to detoxify through several of the well-established pathways in the human body that are designated to detoxification, then in that child, with the unfortunate circumstance of innately poor detoxification, plus whatever is the environmental insult, plus just that right time or wrong time in brain development or just overall physiologic development, we start seeing that we can see far better associations than we ever did before. So, to be more specific, one of the pathways that I study is, and this is where CNVs come in, what if I told you, Kristen, that one of the most important genes in our body, the gene family responsible for detoxification, there's a family of genes known as the GST family. So these genes, you can imagine the detoxification, the human body doesn't leave detoxification or treat it very lightly. And it doesn't leave it, we don't leave detoxification to a single gene. There are multiple pathways, let me be clear. There are multiple pathways through which the body can detoxify itself at the organelle and cellular level. This is the beauty of the human body. Having said that, there are certain pathways that are much more important and radically important than others. And one of these pathways is the pathway that is handled by this GST family. Now, the GST family of genes, there are more than three, but the three major GST genes, they're known by the suffixes T1, P1, and M1. So you have the GST T1 gene, the GST P1 gene, and the GST M1 gene. And what is remarkable, Kristen, is, so these genes are extremely similar to each other. They will make enzymes, each of which can do an extremely similar job of detoxifying the body. But each of these genes are distributed in different places of the human genetic makeup, meaning GST-M1, for example, is found in Chapter 1 of our operating manual. GST-T1 is found in Chapter 22 of our operating manual, and that's an actual analogy. Now, we should have 2 copies of each of this gene. The T1, GST T1 version of this gene, Kristen, can be completely deleted. There are people amongst us, by the way, some of them perfectly normal, who don't have the T1 gene. It is estimated that about 15 to 20% of European Caucasians, people of European descent, don't have that gene. Now listen and take a moment to understand this. It means these individuals do not have the gene that makes that enzyme. If we look in their bodies, not that we're going to find an inefficient version of the GST-T1 enzyme, we're not going to find it. Now, of those three GST family members, T1, P1, and M1, the T1 is by far one of the most important. It is, as I fondly call it, it is the first responder in the body when there are toxic spills. When there's a toxic spill in the body, the T1 is the hazard waste removal crew that shows up on the scene. When you don't have the T1, as is found in 15, 20% of individuals, depending on ethnic background, okay, you still have the P1 and the M1 as your backup. This is number one. This is why people who don't have the T1 may be perfectly fine, so long as they have those backups, and so long as they did not get exposed too much to the toxins that this family is required to get rid of. However, what if I told you then that, believe it or not, there are 5% of individuals who don't have the T1 and don't have the M1, neither. Now here we go. We've got this family, one of the most, if not the most important detox families in the body, and I've just told you that 5% of individuals thereabouts, depending on ethnicity, may not actually have this combo. Now, does that mean that those 5% of individuals will have autism? No. But all of a sudden we begin to understand, if those individuals at critical stages of embryonic development, at critical stages of postnatal toddler development, if those kids were subjected and exposed to toxins that the T1 and M1 genes were responsible for, and those toxins get into the body, and now they cannot be cleared near as efficiently as the individual who had the genes. And if in those children we have the MTHFR variation and other things, now we begin to paint a picture to really understand what is this functional motif happening in the child. I will stop there. Do you understand how we should really be looking at this?
Absolutely. I feel like I don't understand why, to be honest with you, our typical, when we go for our physicals, like why aren't they doing some type of DNA testing like this to kind of be able to help us understand our bodies better, understanding more of a precursor of, potentially things that could be something we should be considering or looking out for in our DNA. It seems as though this technology is here, but I don't feel like we're using it to the best of our abilities in these days.
I could not agree more. Let me give you an example. Kristen, you're a mom, a beautiful mom. My wife and I, we've got 3 beautiful children. Which parent in our generation hasn't used baby Tylenol or children's Tylenol for our children, acetaminophen? And I'm sure, and I know well in the U.S., the FDA and other regulatory agencies have started to mandate that acetaminophen, Tylenol, anything above 250 milligrams per capsule or tablet, now comes with this warning of liver toxicity. It is because when acetaminophen, the active drug, that is an analgesic, antipyretic, for pain, for fevers, all of the things that we use, we all, as families, we have this in our cabinet for our children and for ourselves as we get older. Okay? When acetaminophen is taken, the first thing that acetaminophen is metabolized into, as the body is breaking it down in order to flush it out of the body, which happens to any medication we take, the first thing that acetaminophen is broken down into, simplifying it, is something called NAPQ. NAPQ, N-A-P-Q shortened, is a potent, and let me underline, potent toxin absolutely established, which is why the warning labels are now being put. We've always known this, by the way, because the thing is, even though NAPQ is so potently toxic to the body, horrible things it can do at high levels in the body then has the ability to, so we take acetaminophen, yes, we first break it down, yes, NAPQ is produced, yes, NAPQ is horrible, but then NAPQ is then further metabolized, neutralized, and voided from the body. So, okay, fair enough. That's why we can still, generally speaking, use acetaminophen at normative levels and not have to worry too much. But hold on. The gene responsible, primarily responsible, this is, again, I'm simplifying things here into simple pathways, but one of the most important genes for metabolizing acetaminophen, for breaking it down like I first said, is a gene called CYP2E1, CYP2 Edward 1. We should have 2 copies of this gene. CYP2E1 breaks down acetaminophen and turns it into NAPQ. What if I told you that there are people, and one of the regions where this phenomenon is well documented are people from Dutch, Northern, Germanic ethnicities. They don't have 2 copies, some of them, this is obviously not the entire population, they don't have 2 copies of this gene, they've got 3 copies of the CYP2E1. Now, when you have 3 copies of the CYP2E1, literally, those individuals are producing more of that enzyme. Those individuals are more rapidly converting acetaminophen into that goober-toxic NAPQ. Now, by the way, what is the gene largely responsible for then taking the horribly toxic NAPQ, further metabolizing it, detoxifying it so that it is finally not toxic, so that we can finally flush it from the body? GSTT1. So wait a minute. What happens to the person, simply, what happens to the person who simultaneously has 3 copies of CYP2E1 and happens to be the person that does not have the GSTT1 gene? By the way, those individuals exist. And by the way, those are the individuals that have it. And the thing is, Kristen, it is so easy to determine if you are that person. And those individuals are the ones for whom acetaminophen is a particularly toxic thing to put into the body. That information is there, you see, and that we have some scientists saying, well, but the CYP2E1 variation, having the extra copy is documented and not everyone is ill from it. And independently, the GSTT1 deletion has been documented and not every such person is ill. That is the fallacy of clinical trials, as I explained earlier. It is when we don't apply a functional understanding, it is when we don't apply the understanding that CYP2E1 works in tandem with GSTT1, and if you happen to be the person that had that unfortunate combination of an extra copy of CYP2E1 and 0 copies of GSTT1, which by the way happens, you now become a person who de facto are extremely sensitive, without you even knowing it, to acetaminophen, because when you take acetaminophen and you're that person, your body is rapidly producing NAPQ, and that NAPQ, which is so toxic, is sticking around the body much longer because you don't have the GSTT1 gene to clear it out. This is just, acetaminophen, Tylenol, that is so commonly used. Can you see how important this is, Kristen?
Absolutely. Even with my son, he doesn't take Tylenol anymore, but he did for the first 2 years, 4 months of his life, before I learned other ways. And so, I know many families listening, they were in the same boat. Some families might be listening that, aren't aware about this. I know other speakers we've had on the summit talked about the dangers of Tylenol, for our kids. Mm-hmm. And so I think that, I feel like we're coming together, but at the same time, I feel like we need to get that information out because there's still so many families out there that are not really, understanding the importance of really of the genes, and they're not really understanding the importance of what we're giving and putting into our kids' bodies,
Absolutely. And again, this is to make sure to be clear. We're not saying that acetaminophen and Tylenol will necessarily be toxic or dangerous to every child. I mean, frankly, every one of us, child or not, will do better to have less of this in our bodies. That's just a general. No one can be faulted for making that statement. I mean, it's just clear that we don't want as many of these things in our bodies. However, it is to say that it is an absolute disservice to the medical field that we don't appreciate that some individuals are much, much more affected by these things than others. And if that is happening in our child, and if we could know that a priori and simply be equipped to make better decisions, then to prevent that, or to say that, well, a clinical trial shows that this is not relevant or that's not relevant. I mean, there's so many examples, Christian. And again, I probably will end where we started, and that is, this is the power of functional genomics. It is that genomics and genetic testing must transcend the overly gimmicky, simpleton, simplistic way of saying, here is one gene, here's what it means. We are not some compilation of individual genes doing their own jobs. We are the compilation of thousands of genes working together in harmony. And so we must look at how the genes work together if we are truly to understand when things become disharmonious, we understand what the real cause is. I think that's a good place to summarize, would you say?
Absolutely. And, I have a couple of things I want to kind of talk a little bit about the customized supplementation that you guys are doing. But before I do that, I was reading in a paper that you have, it talks about it is believed that, it's so much better, basically genetic causes could be better criteria to have better translate into better management of the patient through more accurate expectations and better counseling or therapy options. And this is for autism, and because there's just so many different versions of autism, right? There's a spectrum. But what I loved about this, though, and again, I just summarized that, and your paper, if people ever get a chance to read it, I think is really informative. However, one thing, we only have a little bit of time to talk about the customized supplementation, but I know the families listening right now, it's always so tricky, right, when you're trying to figure out what supplements to give your child. And sometimes you say, okay, I give this, whatever it is. Let's say it's vitamin C or a multivitamin or whatever it is. You don't know if that's getting into the child's body. Sometimes you do blood work, and it takes 6 to 8 weeks to get the blood work back. And, then they say, oh, well, this level maybe is a little low, this level is a little high. But then, again, that was a 6 to 8 weeks of not knowing, and then even that's not always 100% accurate. And often, I know many families listening, we give a whole bunch of stuff, and then we're told by the doctor we're seeing that it's not absorbing into the child's body. And honestly, it feels as though you're buying sometimes things that you're just pouring down the kitchen sink is what it feels like, because you might as well have done that because it's not absorbing for whatever reason into that child's body. What I'm very excited about, which you're doing in your organization, your practice, is you are really figuring out better ways to customize supplementation for those, not just with those with children with autism, but you're doing a huge range with hormones and weight loss and everything else. But today we're really just talking about those with autism. And, let's talk about that importance. And, would you agree it's kind of like pouring things down a kitchen drain if our children aren't absorbing things we're giving them?
Absolutely. I'm a firm believer in, first and foremost, we have to understand the importance of proper nutrition. The human body did not develop over millennia and thousands of years. Our forefathers existed before supplements. This is no two ways about it. So nutrition is hugely important. However, when we're dealing with a child, depending on where they are in the spectrum, as many of us understand, it's difficult even for these children simply to engage and have the broad range of nutrition at the whole food level that would be appropriate for their bodies. So we can't look at those parents and those children and say, well, supplements don't work, you've got to have the child eat. What if the child simply is not even eating or able to eat at the level that will be healthy? So number one, I just wanted to say that for many years, I had many supplement companies coming to me asking to, have my name be partnered with supplements and use things, and I resisted. I never wanted to be a genomicist who seemed to say, well, I'm selling supplements, God forbid. However, I have to come to terms with the fact that there are times and places where we do need supplements, as is classically the case amongst our autistic children, simply because they're not eating the fullness and diversity of what might be considered a healthy diet. And even if they were, because there are metabolic imbalances in the body, this is the beauty of where modern medicine comes in, the beauty of modern nutrition and nutrients, that we can understand that there are certain pathways that might need to be fed more or less in a given child. So now to answer your question, the only way we're really going to understand which of the things any one of our children, autistic or not, on the spectrum or not, really needs. Do they need it? What do they need? How much do they need? That will return to that child's innate, unique genetic makeup. It's plain and simple. Now, it is still a fairly young field. It is a field in which we have to be careful. There's a lot being said out there. We have to take a conservative approach, especially when we're dealing with children. Okay? We must be careful. But there's no two ways about it, that there are genetic findings that help us to appreciate if certain things are going to be absorbed better or less. Do we need a little more of it, less of it? So that now these parents, they're dealing with so many other things in their life. The last thing they should be worried about is, am I giving my child the right amount, the right variety of supplements? Given that that child is not eating a full meal or a diverse meal, we should be putting so much of our effort and attention in trying to help that cause, and that's why I finally put my name to this endeavor that we have to do precisely that. Because I've just seen too many families just going, am I giving my child enough? Am I giving my child too much? How could I figure out if I'm wasting my money? That's what this endeavor is about, Kristen.
And I love that, and I know that you guys are doing something fabulous and just so generous to basically our community from the Autism Hope Alliance, and so you guys will be discounting testing for them, and then there's going to be, and you're limiting that to 500, which is beyond generous. So if and when we learn more about that, we will definitely spread that word. So for those listening, if you are interested in that, please email the Autism Hope Alliance, and we'll get that information. But you probably will already have heard about it by the time this is going to be aired. So, but again, you guys will be offering tests, 500 of them, to our families that we feel that are would be qualified and in need for those. So we're and again, those would be at a discounted rate. And then you guys are also going to be doing something. I know this is really new, and but you guys are going to be doing a subscription cost to aim to produce supplements for the autism community based on their actual need. Right now, I know you guys are talking around in the $69 a month mark, and again, guys listening, that may change a little bit because this is as new, but that's what they're aiming for. You guys are really making, I think, a very great contribution to what our families need. And again, for those listening, this is just another option that you can consider for your family. We at the Autism Hope Alliance love to give almost like a menu of just different things we learn about that we want to share and spread that word. Because I know for me as a mom, back in the day, 13 plus years ago, my son is going to be 15 here in a couple of days, but 13 years ago when I got that diagnosis, I was so lost and not knowing where to turn, and I would have loved to be able to have a menu to kind of say, hey, let me look into that, and if that feels right, I want to try this, or let me look into this, or maybe I don't want to look into that right now, but I now at least know it's there, and I can look into that later. And I feel like we know so much more now, and there was nothing like I've never seen anything like this even right now. So this is still something new, and I think it just promotes so much hope that, there's people like you and your organizations out there that are basically saying we're not giving up on our kiddos, and we're going to keep trying to make it easier for our kids and keep trying to make it better for our children. Do you want to add anything to that? Did I miss anything in that?
And that was just so wonderfully said. And I would just say to your viewers, well, to your listeners, I'm sure they already know this, that they have in you, Kristen, just one of the most passionate and wonderful individuals representing their needs at the AHA, the Autism Hope Alliance. And when we met you as a company and as individuals, every single member of our company, from our CEO to myself and others, every single one of us are parents of children. And when we met you in Anaheim recently, and we had a chance to speak to you, thanks to our CEO, he pulled me aside and he said, Monsoor, whatever it is we can do to help this group, having met that mother, speaking of you, Kristen, having seen your passion and how much you give of yourself, he made the executive decision to offer those first 500 tests, literally at cost. So I guess to your listeners, this is a lot having to do with you, Kristen, and having to do with what you and how you represent your families. It truly moved us as individuals, researchers, and as a company, and we felt that we wanted to give this back to the community. I mean, forgive us, we had to draw the line at some point because it's a fairly substantial undertaking to offer it just at cost, but we will continue to give and provide whatever service your community wants from us, and we look forward to doing so.
That is just so wonderful and beautiful, and it's just an honor to meet you guys as well. And what is the website that is there a website that you guys want to share to let people know more about you guys?
Indeed, the website is obviously www. And it's a play on nutrients, a fun play, but it will become apparent. Utrients, Youtrients, Utrients.me, and we chose the .me instead of the .com because obviously I think you can tell it is what it is under the moniker, it's under the theme of your DNA's supplements. We're really and I know that these terms are often over-bantered and overused, but we're really revolutionizing this field. We're saying that we're for the first time at the person-to-person, individual level, we're taking the DNA makeup of that individual, having clinicians, skilled clinicians, look at that DNA, look at the variations that we think we know something about. And by the way, guys, this field is always growing. When you never feel that you understand everything about the MTHFR gene or the SHMT1 gene or the GSTT1 gene or all of these things that can be implicated in how your precious child's body works. Because every week there are new publications, there are new findings. So what we're saying is we have the ability to record some of the most noteworthy variations, whether they're SNPs or CNVs, and we then have the ability to infer based on thousands, hundreds of publications to say what might be a better composite for the nutrient needs of that child, and that's what we're doing. You can find it at www.utrients.me. I have never put my name or tried to seem commercialized, and even right now saying it, no matter as proud as I am, I cringe a little pointing someone to a website. However, many of my colleagues in the academic, in the research, in the clinical field finally said, Dr. Monsoor, you have an obligation to take all of the years of the work that you've been doing, and you do have an obligation to try and start making this and putting it towards something that benefits families beyond those families that come to see me in person, which of course is my day job. I do clinical consults at many different clinics, and I give lectures across the world. So that's the website, Kristen. Obviously, we are here. Obviously, I think for you, being a representative for your community, any things that you filter from your community, you can bring them to us, and we'll do our best to answer and provide services that hopefully change and benefit the lives of the children that we're dealing with.
Thank you so much, Dr. Mohammed. Is there anything that you, last quick message that you want to give to families that are feeling helpless and a little bit scared and a little bit lost? And, is there any message that you might want to leave them with?
First and foremost, thank you for even having me. To those of you who have listened to me or who will listen to me, honestly speaking, I am the one that benefits speaking to you. Every single family that I meet, every single child that I look at and speak with and evaluate, I come away a better person. I'm the one that, I simply can't put words to thanking you for giving me the opportunity to do this. As far as the parents are concerned, I too am a parent, and, they're grown up, they're in their 20s to 15. When they get a paper cut, it still bothers me, literally. So I can't imagine when a parent is dealing with something where their child, they're not sure if their child is hurting, what is going on, because that child may not have the means to explain what is going on within them. So I guess the message to you is, as a parent, I understand that, and it is with that perspective, that passion, that compassion, that I would say, through intelligent genetic testing, what you can afford yourself is you can begin to understand that this is not a mistake that you made. This is not something that you did wrong. These are things, these are variations in our children as human beings that, with the unfortunate circumstance of the unfortunate timing of things. And so there is hope. There's hope to understand why something happened in your child. And that's the first step. I always tell parents. Parents will say, well, Dr. Monsoor, what does genetic testing afford me? What it affords you, if done well, for some families, this is not going to be for everyone, if done well, the genetics becomes the epiphany that you at least are able to stop looking for skeletons in the closet. You have that epiphany of saying, okay, I now understand what is happening, or at least I begin to understand more of what is happening in my child. And from there, we can begin the journey. But it is so important to understand, because you take 10 children on the spectrum that appear to be affected to the same degree, and therefore the logical thing would be to try to treat these 10 children the same, whereas in fact there may be completely different pathways that are affecting each of these children, culminating to look like the same outcome. So that we as parents can equip ourselves to begin to understand at a more individual level. That's the hope that we want to offer. There's a lot of work still to come. There's a lot of, unfortunately, misguided statements out there, both within the scientific community and those that negate the scientific community. So stay positive. Use the type of services available to you. And you have, as parents, never underestimate your own gut barometer of what is good for your child or not. No one can replace that. You're the parent. You're blessed with being that parent. Use that inner sense that you have if this is something that will be beneficial. And if you think it is, then so be it. And if you think not, then also so be it. That's the message I'd like to give.
That's so beautifully put. And, for all of us listening and, out there, I feel like we have just learned so much, and you were just so compassionate in how you delivered that, and you were just so, I feel just so easy to be able to understand the way that the message came across. And I think we've come away learning so much more. So thank you I know you're so busy, and thank you so much for taking the time and really helping us. And thank you guys all for listening. Until next time. Bye, guys.